want to amplify the DNA so that we can work with it. In other words, use restriction enzymes on it and do something else, like subclone it, as we'll see in just a minute. But we can also use PCR as a diagnostic tool, right? So for example, I don't know that it's so common anymore, but back in the dark times, right? In order to go anywhere, you had to get these PCR tests for COVID, for example, right? Well, how does a PCR test work? Well, all a PCR test is doing is testing for the presence of some viral DNA. Okay, and how do we do that? Well, for example, if we want to test for the N1 gene in COVID, all we have to do is design a primer, a forward and reverse primer, okay, that will anneal specifically to the DNA sequence of the N1 gene of COVID. Right? So if you take your swab of your nose, okay, what's going to happen if there's any virus DNA in there at all and subjected to PCR, even if there's only 2 or 3, well, now we get amplification, we can detect that. So 2 or 3 copies of the gene is not enough to detect, but now we amplify it millions or billions of times, now that is enough to detect. Right? So if we get a positive amplification using the N1 forward and reverse primers, that means that there is N1 DNA there, which indicates that there is COVID there, or COVID DNA there. Right? Now one of these is not enough. Right? So we have to use, for example, now we test for the N2, we test for N3. Right? So we test for many of these different viral DNA markers, and if they come positive, in other words, if we get amplification, that indicates that the primer is bound, the PCR was successful, so that means in the original sample, there was some viral DNA there. If we don't get amplification, that indicates that the primers didn't bind, because they didn't have anything to bind to, and so we won't get amplification, that would be a negative result, right? Now, the reason why PCR tests were preferred is that PCR is so sensitive. Again, you don't need very much starting material in order to get a very large output, right? We're amplifying this exponentially. This is different than, say, the at-home kits, which were ELISAs, you remember from chapter or something else, right? This requires a larger amount of initial sample in order to bind all these antibodies, okay? Here, this requires very little, right? So this is much more sensitive. And we can also use PCR as a forensic tool, okay? So beyond medicine, we can use this forensically. And this relies on this phenomenon here, that in all of our genomes, we carry different markers, which are called VNTRs, variable number of Hammond repeats, okay? And on each chromosome that we also have, at that same marker, different number of repeats, right? So what's a repeat? It's just a sequence of DNA that repeats over and over and over for no discernible reason, right? It's just a quirk of evolution, something left over from millions of years ago, okay? And how is this used? Well, if we look at, for example, individual A, it's got 2 chromosomes. For VNTR1, the first marker, he's got 3 repeats on chromosome 1, and 1, 2, 3, 4, 5, 6, 7, 8 repeats in chromosome 2, right? So if we design a primer that blanks, right, this will always be the same, no matter how many repeats we have in the VNTR, right? This sequence will always be the same, and this sequence will always be the same. Well, now, if we do PCR using these 2 primers, we should get 2 fragments. One fragment that is this long, and one fragment that is this long, right? And the same for VNTR marker 2, right? Well, now we should get a fragment that is this long, and another fragment that is this long. Same for VNTR 3, and so on, right? To get a positive ID, you need about, I think it's up to 15 markers now, okay? So here's just showing 3. So individual A, okay, will have a pattern with VNTR1 having this size and this size, VNTR2 having this size and this size, and VNTR3 having this size and this size. Individual B and individual C obviously have different numbers of repeats, even though, for example, in VNTR2, on chromosome 1, they both have 3 repeats, right? On chromosome 2, they have a different number of repeats, right? So this helps to identify a person. It tells us how many repeats at each marker on each chromosome an individual has. Now, the chances of 2 individuals having the same number of repeats on both chromosomes is so small, especially when you look at all of these different markers, that it's used to exclude basically anybody who's ever lived, right? You get a match of 1 in, tens or hundreds of billions, right? So PCR can be used clinically and forensically. Now, recombinant DNA is probably one of the biggest advancements or discoveries in since the printing press. Right? So, how does recombinant DNA work? It allows us to synthesize and modify, synthesize DNA rather, and modify organisms at a molecular level. Right? In fact, as we'll see here in just a second, insulin, right? Insulin was first produced not chemically, right, but it was produced using these techniques, recombinant DNA. All right, so the tools of recombinant DNA technology have been used to identify disease-causing mutations. We get polymorphisms, right? This is instances of genetic variation, and restriction fragment length polymorphisms, RFIPs, right? Polymorphisms within restriction sites that change the size of DNA fragments. So this is like what we saw when we looked at those two viruses on slide 1, right? Or slide 2, I guess. So there's a way to correlate mutation with the occurrence of a disease, right? So let's look at recombinant DNA. Or recombinant DNA technology relies on a vector, right? And a vector is a DNA molecule that can replicate autonomously, right, in the appropriate host organism. So essentially what they act as is an accessory chromosome, right? Specifically in the case of plasmids. We'll get to plasmids in the next slide, right? The choice of vectors for cloning in E. coli, right, which is probably the most easy to use organism or host for recombinant DNA technology, include plasmids and viruses, specifically bacteriophages. Now, plasmids are, like I said before, essentially just accessory chromosomes. Recall that the genome of a bacteria is circular, right? That's how they achieve supercoiling. We talked about that last week. Plasmids are also circular, okay, but they're much smaller. And the bacteria, E. coli in this case, treats that small circular piece of DNA, it treats that plasmid as it would treat the chromosome. So it's replicated independently of the chromosome, right? It just thinks that there's an extra chromosome, okay? Bacteriophages are a little bit more complicated. I'm not going to spend too much time talking about bacteria, or rather bacteriophages, but they allow us to store larger fragments of DNA than what is possible on a plasmid, okay? So, inserting DNA into vectors, right? First we have to cut the vector by using a restriction enzyme to create cohesive ends. We'll see that in just a second. Kind of have an idea of how that's going to happen already. We can add linkers, if necessary, to yield cohesive ends. Okay, so we can add sequences of DNA to yield cohesive ends if there are none available. And then we insert the DNA fragment with the cut vector using DNA ligase. Right? So let's see how this would work out. So again, here's our restriction enzymes. These are how they work. Again, they're going to cut the DNA, yielding 5' or 3' overhangs, not blunt ends in this case. So what's going to happen, again, here in the case of BAM-H1, we're going to end with a 5' overhang on both strands. Here we'll end with a 5' overhang on both strands. Here with a 3' overhang, and so on. These, again, 5' and 3' overhangs are called sticky ends or cohesive ends for reasons that are shown right here. Okay? Let's say that we want to put this piece of DNA into this piece of DNA. Right? So we can consider this to look like this. Okay, why is this always blank? So again, here's our piece of DNA that we want to cut out and insert into a vector, okay? So let's just focus on one side for now, right? So let's just focus on this side over here, actually. Let's focus on that side over there. All right, so what's gonna happen? Here, if there's an EcoR1 site, here, and if in our vector, there's an EcoR1 site here, well, now we have compatible sticky ends, right? Two cohesive ends generated by the same enzyme are always compatible, meaning they can base pair together. And we can see how that is here. So in our example, right, this blue would be on this side of the cut, and this blue would be on this side of the cut, right? And the same for the pink, right? Let's say that the pink is the vector, so the pink on the left in the middle there is going to be on this side of the cut, and the pink on the right is going to be this side of the cut. So what do we want to do? We want to anneal this side of the cut in our insert to this side of the cut in our vector, right? And that's what's shown here, right? So we want to anneal this into this. Okay, so let's ignore these two for now. So what happens? Well, here again we have a 5' overhang. Here we have a 5' overhang. Now because they're compatible, they were generated by the same restriction enzyme. Well, this can base pair with this, can it, right? TTAA is going to base pair with AATT, right? So we get a base pairing here. So we've now annealed these together, right? So we've inserted this piece into here. Now we can do the same thing on the other side, right? We can say, well, let's say that there's again an EcoR1 site here, and an EcoR1 site here, okay? Now we want to anneal this side of the cut with this side of the cut, and what we see is that is this blue piece annealing with this pink piece, which we see over there, right? So this would be like what happens over here, and this is what happens over here. So now we've got both sides annealing, and effectively what we've done is break open our plasmid and put our insert right into the middle of it, right? So this situation is what's happening over here. So let's say that this is what's going on the left, and this is what's going on the right over there, right? So now we've got our piece of DNA into our plasmid vector. Now, there's one thing left to do here, right? All we've done is form base pairs here, but we've only got base pairing between 4 bases, so this is not very strong. And we have a break in the backbone right here and right here, right? So this backbone stops and this backbone begins, right? So there's a break in the phosphodiester backbone. In order to fix this, we need to add DNA ligase. DNA ligase is a ligase that's going to ligate 2 different DNA fragments together. And now we'll have a contiguous DNA backbone here, contiguous DNA backbone here, held together by these base pairing interactions. Okay? This happens on both sides. Now the downside of doing it this way, using the same restriction site on both sides, is that this insert, right, the piece of DNA that we're inserting into the plasmid vector, can be inserted the other way around as well, right? Because both sides are going to be compatible. So we can get insertion in one direction, or we can get insertion upside down. How could we get around this? What could we do? What could we modify here in order to make sure that we get insertion only in the correct direction, so we don't get insertion upside down?
Use 2 different restrictions.
Yeah, we have to use 2 different restriction sites. So we have to either find a different restriction site or insert a different restriction site here. And the same thing on our plasmid. So now, this BAMH1 site is going to generate a compatible cohesive end with this BAMH1 site on our plasmid vector, but this BAMH1 site is incompatible with the EcoR1 site here. So it can't anneal backwards, it can only anneal forwards, in the same logic for the EcoR1 site. In fact, this is how most plasmid vectors are engineered, right? So plasmid vectors, we typically just don't find in the wild anymore. They've been engineered. They have a few common features. They have a few common features. First, and perhaps most importantly, they have what we call the multiple cloning site or polylinker. The multiple cloning site consists of many different restriction sites, right? Anywhere from between 10 to 20 different restriction sites here, corresponding to many different restriction enzymes. So in our original vector, right, we might have one that's got a BAM site over here and an ECO site over here. So when we cut it, now we can put our BAM site over here and our ECO site over there, and we can control the direction in which we insert the DNA, right? So this polylimber or multiple cloning site contains an engineered sequence that contains many different restriction sites, okay? It also contains what we call the ORIC. The ORIC is the origin of replication, because then remember, the cell is going to treat this, the bacteria is going to treat this as an accessory chromosome, which means that it needs an origin of replication just like a real chromosome, right? So we have to make sure that this is there, otherwise, whatever we give the bacteria is it. It's not going to make anymore. And the whole point of this, right, is that when we give the construct, right, the modified plasmid to the bacteria, they're going to maintain it, they're going to replicate it, and we can just go back and harvest it when we need. We also need some form of selection, usually in the form of some sort of antibiotic resistance like ampicillin. Right, so an ampicillin resistance gene or a kanamycin resistance gene or chloramphenicol or what's the other one? But anyway, some sort of antibiotic resistance. The reason for this is that when we go through the process of giving the plasmid that has been modified to bacteria, that process is called transformation. That process is very inefficient. Only about less than 1% of the bacteria that we present this plasmid to are going to accept this plasmid. The rest are going to go on living their happy lives. So if we try to grow up that population of bacteria, we're going to have a minority of bacteria that contain the plasmid. And this is a problem because now the unmodified bacteria will outcompete that modified bacteria, and eventually our modified bacteria carrying our construct will die out, right? So we'll lose it very rapidly. By having this ampicillin resistance gene here, well, now we can grow that population of bacteria that we've presented our plasmid to in a media that contains the antibiotic, in this example, ampicillin. All of the unmodified bacteria will die out, and the only ones that will be able to survive are the ones that are carrying our plasmid, right? So now we'll get selection. This is a selectable marker. So these are the three hallmarks, really, of a plasmid vector. We need a multiple cloning site so that we can actually insert our gene in the origin of replication so that the bacteria can replicate that and maintain it. And we need some sort of selectable marker. Now, in addition to transformation itself being inefficient, meaning that we need this selectable marker, the process of subcloning, which is, putting our piece of DNA into the plasmid, is also inefficient. So now we have two sources of inefficiency. We were able to deal with the first by having this selectable marker, which means that all of our bacteria should be carrying the plasmid, but the plasmid that we presented to the bacteria is not guaranteed to be the one that we modified, right? So what we'll actually have are two populations of plasmids, right? So we'll have a plasmid that has the multiple cloning site, and we'll have a population of bacteria, or of plasmids rather, that contain our insert, right? So this is plasmid A, let's say, and this is plasmid B. Plasmid A, right, was not transformed. It was not modified, not transformed rather. It was not modified. So it does not contain our insert, right? This is just an effect of the inefficiency of the process of subcloning. So we'll have some unmodified plasmid, but we'll also have modified plasmid, okay? Now, when we transform, so we have these together in our reaction vessel, little tube, right? And we present this to the bacteria, and some of them will take it up. Now, both of these have the selectable marker. Right? Both of them have this ampicillin resistance gene, or kanamycin resistance gene, whatever. So now when we grow our bacteria up in this antibiotic, both will be able to survive. If they took up this plasmid, the empty plasmid, well, they'll be able to survive because they still have the resistance gene. If they have the insert, they'll also be able to survive. So we'll have now a population which does contain a plasmid, but we're not sure if the plasmid that they have actually contains the insert. So how do we get around this? We get around this using what we call blue-white screening, all right? And blue-white screening relies on this gene here, LAC-Z, or beta-galactosidase. And what beta-galactosidase does in a bacteria is essentially it takes the disaccharide, lactose, rather, takes the disaccharide lactose and it hydrolyzes it into a glucose and a galactose. We'll talk more about that in chapter 11. But essentially it's necessary for lactose metabolism in bacteria. Now as it turns out, right, beta-galactosidase doesn't actually care if there's a glucose here. All it cares about is there a galactose here, and is it bound to something else? If it is, I'm just going to break that bond. It doesn't care if there's a glucose after. So we can, instead of giving the bacteria lactose, we can give it this compound here, 5-bromo-4-chloro-3-indoleal beta-degalactopyranoside. That's a mouthful, so we just call it X-gal. Okay? This has been modified, so what we have is a galactose here and a dye bound to it. If beta-galactosidase is present, right, it will hydrolyze this bond, releasing a galactose and the dye, right? So now if beta-galactosidase is present, we'll have a lot of this dye. This dye oxidizes and dimerizes to form a blue precipitate, right? So the cells that have functioning beta-galactosidase will appear blue because they're able to undergo this reaction in the presence of X-gal. Bacteria that don't, however, will remain their natural color, which is kind of a pale yellow. So how do we use this? Well, what if we put the polylinker, or the multiple cloning site, inside of the beta-galactosidase gene? Right? So we put it in the coding sequence of the beta-galactosidase gene. So notice here, the multiple cloning site has many different restriction sites, one for MU3, PA1, there is EcoR1 over there, XPAL1. So we have many different restriction sites in the polylinker, but the polylinker itself is in the beta-galactosidase gene. So now, if our cloning was successful, what we'll have is something like this, right? Here we have a green insert in the middle of the beta-galactosidase gene, which means that we've broken the beta-galactosidase gene, so these bacteria won't make any functional beta-galactosidase, and they won't be able to produce that blue precipitate, right? If our cloning was unsuccessful, then we'll have an uninterrupted beta-galactosidase gene, right? Which means that we'll have functioning beta-galactosidase and the cells will turn blue, right? We can see that over here. The white colonies do not contain any beta-galactosidase, so they don't turn blue because they've got our insert, and the blue colonies do have functioning beta-galactosidase. We can see an example of that, just more on that right here. So this is what a plate actually looks like of blue-white screening, right? So we take our bacteria, we transform it with our inefficiently subcloned plasmid, right? They all contain ampicillin resistance, let's say, so they'll all be able to grow on a petri dish that contains growth media plus the antibiotic, right? So now we know we have a population of bacteria that contain either this or this, and in order to distinguish between this and this, right, we know that the empty plasmid containing bacteria should appear blue because they have functioning beta-gal, whereas the white colonies are white because they contain our insert, disrupting the beta-gal gene. So here we have all our blue colonies, which are really easy to see, right? These are a brilliant blue, and our white colonies here. The white colonies contain our insert, so it allows us to distinguish between ones that took up the empty vector and the complete vector. Now, that's fine if we want to just subclone a piece of DNA to work on it or to maintain it. Okay, but very often what we want is to actually express that gene or that piece of DNA, so turn it into a protein. So all we really need, okay, for a plasmid, the bare minimum, again, is that origin of replication, antibiotic resistance, and a multiple cloning site. If we want higher efficiency, we add a reporter gene, which is in this case that beta-gal, right? This allows us to make sure that we're getting the correct thing, right? So now we have 4 things that we would need. But if that's it, there's no way now to do any biochemistry with that piece of DNA, right? So we can't transcribe it, we can't translate it. And so what do we need in order to actually turn that piece of DNA into a protein? So for this, we need a transcriptional operator sequence, or a promoter sequence, okay? A promoter and terminator sequence is going to drive transcription. So just upstream of our, what do we have here, polylinker, right? So here it's going to be transcribing in this direction counterclockwise. So So we have to be sure that we put our piece of DNA in correctly, so we have to pay attention to which restriction enzymes we're going to be using. In this case now, we have a transcriptional promoter, which is going to tell the bacterial RNA polymerase, hey, bind here, there's a gene I need you to transcribe. And RNA polymerase is going to treat this again, just like the chromosome. It'll bind here, transcribe whatever comes after, right? If whatever comes after contains the correct sequence to be recognized by the ribosome, so it has a Ch.9 RNA sequence we'll talk about in chapter 30, that messenger RNA will then be recognized by the ribosome and translated into a protein, right? So in order to actually express protein from the DNA sequence, we need to ensure two things. First, that our vector has a transcriptional promoter, right? These are typically present in expression vectors. And we need to make sure that our piece of DNA also encodes the necessary instructions for the ribosome, telling it to bind and begin transcription, right? Now, lambda-phage vectors are also useful, okay? There is a limit to how big a piece of DNA we can fit into a vector, right? The vector cannot be too large. If it's too large, it physically can't get into the bacterial cell when we present that vector, that construct to the bacterial cell. So there is a limit to how big we can make them. However, if we do need to insert a larger piece of DNA, we can use a lambda-phage. And lambda-phages are kind of interesting in this way. If you remember, bacteriophages are those kind of weird, spiky-looking viruses that attach to the outside of the cell and inject their genetic material into the cell. As it turns out, about 25 kilobases, 25,000 base pairs worth of information can be just removed from the virus with no effect on anything. Right? You can replace that 25,000 bases with any sequence. The virus doesn't care, and it will continue to function. So inject this genetic material and continue on with its life cycle. So what we've engineered are lambda-phages in which the genome, again, contains multiple cloning sites, so that we can cut out that 25,000 base pairs and we can reinsert whatever sequence we want. And the virus will continue on with its life cycle as normal. Again, remember here, the bacteriophage attaches to the outside of the cell, injects genetic material. At this point, it can enter either the lysogenic life cycle or the lytic life cycle. The lytic life cycle is, as the name implies, it lyses the cell open by increasing the rate of transcription and translation of its viral proteins, generating viral capsules, encapsulating all of the lambda DNA, and then lysing the cell open, infecting other cells. Or it can just integrate itself into the genome of the bacteria and hang out. The weird thing is, once we're in the lysogenic life cycle, we can induce the lytic life cycle. So this is very useful for us. We can add our piece of DNA into the viral genome, allow the virus to infect the bacteria, allow our piece of DNA to now integrate into the genome, and just hold on to it forever, right? Now our fragment of DNA is part of that bacteria forever, okay? As long as we don't induce the lytic life cycle, that bacteria will grow forever and ever, right? Forever and ever, for a very long time. Now, if we need more of that piece of DNA, well, now we can go ahead and induce the lytic life cycle. This will lyse open bacteria, and we can harvest all of these viruses, and now we have a lot of our DNA. So it's a way to maintain DNA. All right, here's more on that, which is fine. Now, why would we want to do this, right? Either using lambda-phage or using plasmid. Well, it can be useful for a variety of things, but it's particularly useful in pharmaceuticals, okay? Say that there's a protein that we want to make, right? There's a protein we want to make. Is there a way that we can make a lot of that protein? We could do it chemically, right? But chemically, it's very complicated, takes a long time. It is possible to chemically synthesize a protein, but there's a limit in length and in efficiency, right? And it's very expensive. So what can we do? Well, it's much easier to just co-opt nature, right? So we're going to hijack a bacteria and make them do it. Bacteria are exceptionally good little protein factories. There's one problem, though, is say that the protein that we want to make is a eukaryotic protein, which comes from the eukaryotic gene, which we talked about last week, have to be spliced and modified. So if we just take that gene, right, we just take that gene and put it into a bacteria, well, the bacteria will very happily transcribe that into messenger RNA and turn it into protein. But it's also going to transcribe the introns, right? And it'll turn the introns into amino acid sequence as well, right? Which will destroy the protein. Right? There's a reason that it has to be spliced. So how do we get rid of the introns before we give it to the bacteria? Because the bacteria has no mechanism by which it can splice. Splicing is specific to eukaryotes. So for this we need cDNA or complementary DNA. And what the DNA is going to be complementary to is not the DNA gene, but instead it's going to be complementary to the messenger RNA derived from that gene, right? So how does that work? Well, we can isolate messenger RNA easily enough, okay? We can use the poly-A tail, right, to isolate it, or we can use another type of affinity chromatography in a way to isolate a specific messenger RNA which contains a specific sequence for our gene, right? However we do it, let's say that we have now, okay, the messenger RNA for our gene of interest. Let's say that it's proinsulin, the precursor to insulin synthesis. Now this, because it's a mature messenger RNA, has already had its introns spliced out, and importantly, it's already been modified, meaning that it's got the poly-A tail on the 3' end, and it's got the 5' cap on the 5' end. Now, because all messenger RNAs have the poly-A tail, now we can design a primer that specifically binds to that poly-A tail. So instead of not instead of, but we can design a primer that's got an oligo T, right? So oligodeoxythymine, right? What's going to happen now? Well, this oligo T DNA strand is going to bind to the poly-A RNA strand, right? We can add reverse transcriptase here. Reverse transcriptase is an enzyme that uses an mRNA DNA, or uses an RNA DNA hybrid molecule as a template, but extends only the DNA, right? So here we extend the DNA in this direction, this primer, and what we have now is a DNA strand that is complementary to our messenger RNA. This is the cDNA, the complementary DNA. Now we can get rid of the mRNA. We don't need that anymore, right? We have effectively that sequence already now preserved in the cDNA strand. We can do that just by alkaline digestion or alkaline hydrolysis. If we increase the pH far enough, okay, it will, as expected, disrupt these hydrogen bonds, but something else interesting happens at high pH. This messenger RNA is degraded, right? So we get rid of that messenger RNA, or RNA molecules in general are degraded, including messenger RNA. That leaves us with only cDNA. Now we need a reverse primer. We need a primer that's going to bind to this side of the cDNA and extend in this direction, so we end with a double-stranded DNA, right? In order for this to happen, we need to add a known DNA sequence, right? We might know the DNA sequence, right? But if we don't, we need to add one that we do. In this case, we're going to just add a bunch of genes using an enzyme called terminal transferase. Terminal transferase is actually very interesting in that it's a template-independent DNA polymerase, meaning that it will synthesize DNA molecules in the absence of a template, right? And it'll do this in a random way. So if we have a primer or DNA strand, a single strand, in solution with a mixture of all 4 deoxynucleotides, it'll just add a random sequence of deoxynucleotides to the 3' end. But if instead of having all 4 nucleotides, we only give it 1 nucleotide, like deoxyguanine, well now, it'll only add G's, because that's the only thing that's available. So terminal transferase with DGTP will now just add a bunch of G's onto the 3' end here. So now we have a known sequence. We can add an oligo C primer, right, which will bind to the oligo G and synthesize the corresponding strand. Now what we have is a double-stranded DNA where this strand is the same sequence as the messenger RNA that we started with. We can now take this, right, we can add our linkers if we need, okay, we can clone that into a plasmid. We can then transform E. coli with this plasmid. If this is an expression vector, now we can induce the expression of this gene because we have a transcriptional promoter there. This will make messenger RNA of this gene in the bacteria. And because the introns are already cut out, what we have is functional proinsulin. This functional proinsulin can then be isolated and turned into functional insulin, right? Which just requires cutting it in one place. And this is how insulin was originally made, right? Not Jonas Salk. Who was it? Anybody know? famous name. Oh, whatever. He sold a patent for $1 to the university, and now it costs a ridiculous amount, obviously. Okay. Now, we can also use recombinant DNA to generate proteins with new functions. Right? So recombinant DNA techniques allow for the introduction of specific mutations in vitro. Right? So we can actually introduce mutations into proteins by modifying the DNA which codes for those proteins. We can introduce deletions, we can introduce insertions, and we can introduce substitutions. And we can do this all using a method called site-directed mutagenesis. Actually, that's very fun to do this. Right? When you do it the first time and you see it work, it's kind of very fun if you're a huge nerd. So site-directed mutagenesis allows the generation of human proteins with a single amino acid substitution, right? We can also use site-directed mutagenesis to generate deletions and insertions. So that site-directed mutagenesis is actually very important, or very potent, right? Point mutation, of course, just mutation where only one base is changed. So let's see how we can introduce a point mutation and then extrapolate that and how we could use site-directed mutagenesis to introduce an insertion or induce a deletion, right? So let's say that we have this piece of DNA, right? This is the parental plasmid, okay? And this parental plasmid contains our gene of interest here, and what we're interested in is modifying this base pair. This CG base pair, we want to turn it into something else, okay? Well, what are we going to do? Well, the first thing to understand is that when we transform bacteria with plasmid DNA, part of their defense mechanism against viruses include methylating all of the DNA in their cells, right? So all of bacterial DNA is methylated. Viral DNA is not methylated. That's how we can distinguish between its DNA and foreign DNA. So after a few generations, okay, all of the plasmid DNA that's being carried by these bacteria are methylated. So that's what we see down here, right? So first thing we want to do here is to separate the plasmid strands, and then we want to add specifically mutagenic primers. Right? So this process is going to be similar to PCR, but we're going to do the PCR intentionally incorrectly. Okay? So we separate these two strands. So we have the dark bottom strand up there, right? We have the light blue strand down here. Now what do? Well, here we add our primer, 5' to 3', which includes a mismatched base, right? So notice here we have A base pair with T, C with G, A with T, so on and so on. But here we have G with G, which does not form a base pair, okay? But that's okay, that's what we want. We want to induce this mutation. And we add a corresponding or a complementary primer on the other side, right? Again, A with T, G with C. Here we have C with C. So this used to be a GC base pair. We're going to turn it into a CG base pair, as we'll see. So what's going to happen, right? Now, we run a PCR here, right? So let's just go through one cycle of PCR. This, remember, this blue strand is circular. Nothing's going to happen there because there is no 3' hydroxyl to extend. The only thing that can be extended is the primer. We're going to allow DNA polymerase to extend all the way around until it reaches back on itself, right? So we'll have a very long product from our PCR, right? It's not going to be circular because there's no ligase, so we'll still have a 3' end here and a 5' end here. They'll just crash into each other, right? And the same is going to happen here. We'll allow this primer to be extended all the way around until it crashes back into itself. Now we have two very long pieces of DNA, dark yellow and light yellow, which should be complementary the whole way around, except for, including the mismatched base, right? So here again we have the light yellow, which was extended all the way around, and we have the dark yellow, which was extended all the way around. Everywhere is complementary, because remember, this is complementary to this, and this is complementary to this, and these two are complementary to each other. These two should be complementary to each other, and they are. See, here we have an A with a T, a C with a G, A with a T. Here now, though, we have a G with a C, right? Instead of a C with a G. Now, again, we still have nicks here, so we can add our ligase, right? This is going to seal these nicks. So now we'll have a full circular double-stranded DNA, which includes our mutation, right? Going from a CG base pair to a GC base pair, right? by introducing this incorrect. But now, again, remember, we still have our parental, right? We have our parental plasmid DNA, right? And we have our newly synthesized mutant DNA. If we just transform this now, again, we'll have a population that contains roughly half and half. Some will be mutant and some will be parental. But remember, the parental is methylated, and it was methylated by the bacteria. Because all of this business here took place in vitro, our product, mutant plasmid, is not going to be methylated because this is not occurring inside of a bacteria. So now we can add an enzyme called DPN1. DPN1 simply just digests randomly methylated DNA and leaves non-methylated DNA alone. So it will destroy this methylated parental DNA, leaving our modified DNA alone. Now we have a population that consists of only our mutant plasmid, and we can go on and transform this. So now again, we've changed a CG base pair to a GC base pair, right? Or we can change a GC base pair to an AT base pair. We can do whatever we want, okay? Now, this is site-directed mutagenesis. Now, site-directed mutagenesis, SDM, can also be used to add an insertion, right? In other words, to insert a piece of DNA. How might we use this method here to insert a sequence of DNA? So say here, instead of changing this to a GC base pair, right? What if instead, between this base and this base, we wanted to add some sequence of DNA? What could we do? Oh, I'm sorry, you had a question before. It was a long time for that.
I could say, like, would the primer still bind if there was like a little bit of unifying? Like, at what point does the primer stop binding?
Yeah, that's exactly right. Right, so we can synthesize a primer that binds the whole way around, but in between